Hello and welcome everyone to CyVerse's Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar is entitled Deploying Scalable Interactive Bioinformatics Analyses via VICE, presented by Peter Rose, Director of the Structural Bioinformatics Laboratory at the San Diego Supercomputer Center. I'll turn the webinar over to Peter shortly, but first some quick housekeeping. Today's presentation is roughly 30 minutes and we'll do Q&A at the end. So please be sure to open the Zoom chat window and type your questions there for Peter, and we'll address them afterwards. Materials from today's webinar, such as the presentation slides, tutorials, and video, will be posted on our wiki page, and you can visit that at any time. I'd also like to let you know about an upcoming training that CyVerse is offering called Foundational Open Skills Science Camp. We call that FOSS for short. It's a camp-style event in June at the University of Arizona intended to give new PIs and early career researchers a solid foundation in free open source computational resources and open science skills and best practices to implement their projects more effectively. Also, I wanted to let you know that our next webinar will be on April 5th, entitled Doing Metagenomics with QIIME 2 Using Jupyter Notebooks in CyVerse VICE, to be presented by Upendra Devasetti, a CyVerse bioinformatician. Please go to CyVerse's events page on our website for more information and to register. And later today, after this webinar, you'll get a CyVerse email with links to the wiki page, our website learning page for more information on the FOSS camp and webinars, and also a brief survey to get your feedback. So without further ado, I'd like to introduce today's presenter, Peter Rose, whose team at the Structural Bioinformatics Lab has built the protein data bank into the only worldwide archive of experimentally determined structures of proteins, nucleic acids, and complex assemblies. The PDB is a key resource in biology and drug delivery, and today Peter will demonstrate how to mine information from it using their application in CyVerse's VICE extension of the Discovery Environment. Welcome, Peter.
Okay, thank you very much, Tina. And I really appreciate the opportunity for us, to present our latest work. So, as you probably know, CyVerse has a new feature. It's called VICE. It's called their Visual Interactive Computing Environment. And it allows you to run basically web-type application like Jupyter Hub, RStudio, or Shiny applications within VICE. And today I'm going to show an example how we use that for 3D structure analysis. And I think this is the first time that actually a 3D structural analysis is run in CyVerse and in the VICE environment. So as Tina mentioned, I used to work for the Protein Data Bank. I ran the Protein Data Bank team here in San Diego for 9 years. So I'm very familiar with that. So the PDB, as you know, is growing very much like any other biological resource. And actually just a few days ago, it achieved a milestone of 150,000 structures. But what is really amazing is that on a per day basis, almost 1.8 million files are downloaded, and that is per day. So one question we're gonna ask later on, what are people doing with all those files that they're downloading? And again, there are almost more than 1 million users that use the PDB in some form. So what we want to work on is data-driven analysis. And what that means, we're not going to look at one protein at a time. We may want to look at an entire protein family, or we may want to look at all the structures in the protein data bank at a time. And there are some barriers of doing that. If you think about a web application, a website, you basically have to do everything manually, and you have a fixed set of options that the website provides. And it's also fixed in terms of what kind of data are being integrated. And more importantly, you have limited options how to output the data from a website. Maybe you can export some data, but the bottom line is we can't really do data-driven discovery manually. This is not reproducible, we can't automate it, and it's also not scalable. So when I worked for the PDB, there were lots of users. They had some specific research problem, but there's no way you can do this manually. It needs to be automated in some way. And what people do, they write scripts or they use, command line applications. And I call this ad hoc scripting, where you're not really a professional software developer, but you just need to, get some work done. You write some ad hoc script and maybe use some legacy applications, you use some non-interoperable file formats. And one of the challenges too is that the protein data bank consists out of 150,000 separate files. So there's also some scalability issues. But doing this type of work the problem is that it's not really reproducible or reusable. This is a one-time thing you do. It's not scalable either. You cannot easily, work on 150,000 files, and it's also not interactive. So we thought there's really a gap, and the gap is for ad hoc analysis. And with that, I mean a specific research problem that you cannot completely solve using, for example, just a web application. So in this chart, you have several axes. This is ease of use. If you have a website, that is obviously the easiest way for a user to get information. On the other hand of the spectrum, on the bottom right, is where you have used a cloud environment or a HPC environment. So in this case, you really need programming skills and so forth. Somewhere in the middle is your own laptop or desktop, and that's difficult too if you're not a specialist in terms of getting the software environment set up and getting the data files. And I think to fill this gap, this is where so-called notebooks come in, computational notebooks. And I will explain more what those notebooks are. In particular, in our case, we're dealing with Jupyter Notebooks. There are other notebooks too. If you're an R user, for example, there's RStudio and so forth. And a notebook is basically a web page. It runs in your web browser, except it's interactive. You can change parameters. And if you know how to program, you can actually change the code on the fly and see that. So the idea here is it looks like a web page, but you can automate it, you can reuse existing functionality, and you can actually reproduce an analysis here. So let's discuss this some more. So how does a Jupyter notebook, first an overview here. So this is a prototype we have set up on Vice. So first of all, CyVerse has two components. One is we need data sets, and they are stored in the CyVerse data commons. So this is the place where we can have the data sets. On the other hand, CyVerse provides an environment for on-demand computing. Built on top, we have built a layer for the analysis of 3D structures, and all this is becoming accessible in your web browser through a Jupyter notebook. Okay, so this is some of our goals that we set for ourselves in this environment. First of all, we really want to be able to reproduce, calculation, and that means we need to define both the software and data dependencies, and we want to do that in a platform-independent way. We want to be able to our analysis to be reusable by others or extendable. For example, having people, change some parameters and rerun the analysis again. Very importantly, we want to be able to share our analysis or be able for others to rerun our analysis. And this is where hosted environments come into place, such as CyVerse that hosts user accounts where we can actually share, analysis with other users or with the public. We also want to have it automatable. That means there are no manual steps, and that requires that we have clean and standardized data sets. And important, we want to have this scalable. To do data-driven discovery, you want to do this in an interactive way where you can easily change parameters on the fly. So we need to be able to deal with very large data sets. And finally, we want this to do interactively. That means we can do some iterative analysis and change things, as we go. Analysis. So Jupyter Notebook is one way of doing it. And here's a nice cartoon that I found on Twitter. So here's Jane. She did some analysis and she wants to share that with the world. So what she does, she creates a Jupyter Notebook. And if you're not familiar with that, a Jupyter Notebook is basically a document that contains description text like a PDF file would do, but it also contains live code that you can execute, and you can have visualizations, in your notebook itself. So a nice thing about a notebook, it combines everything together into a single document. So, and you can rerun this analysis and reproduce it. Now, the next step is you take your Jupyter Notebooks and publish them as open source code, for example, on GitHub or GitLab, etc. You need to make it, you need to publish your software. Finally, you need to set up your software environment, and one way that is often done is through so-called a Docker image, where you define basically a reproducible software environment. And once that is defined, then this reproducible software environment can be run, for example, on CyVerse, and then shared, with users around the world. It can be rerun and so forth. And we actually recently worked on a paper here, 10 simple rules for reproducible research in Jupyter Notebooks. You may want to have a look at that for guidelines, how to really do a reproducible analysis using Jupyter Notebooks. Okay, so here's an example of a Jupyter notebook that does a simple structural analysis. This is actually the first notebook that I ever wrote that does a structural analysis. So it's very simple. If you're not familiar with programming, don't worry about it, but I just want to give you a flavor. So in this notebook, you have descriptive text in here. Then you have code in here in those gray cells. As an example, this last single line reads the whole protein data bank. So with one single command, you can get access to the whole 155,000 structures in the PDB. And in this case, this notebook extracts zinc interactions. What are all the interactions that zinc makes? And in the end, we can show a 3D visualization here. And we list, for example, what are the top 5 amino acids that interact with zinc. So this is a very simple example, but kind of shows you what we can do. This is also reusable. Let's say you're not interested in zinc, you're interested in iron. The only thing you need to do, you change the zinc here to iron and rerun the analysis, for example. Okay, so we have developed an environment that's scalable, and that means we can analyze the whole protein data bank interactively. And as I mentioned, the protein data bank has 150,000 files, and you cannot really do an interactive analysis on 150,000 files. So we developed a file format called mmtf, the macromolecular transmission format, which is a very compact format. So instead of having 150,000 files, we just have very few files. So we can actually run interactive analysis on it. And the entire protein data bank in this file format is only about 9.3 gigabytes. So it's a relatively small file, and that can be processed in parallel using our mmtf framework here. So this is based on 2 technologies. One is Spark for parallel processing and our mmtf format. And on top of this mmtf-pyspark platform, which is Python-based, we have a number of applications, mmtf-genomics, and I'll show you a demo today, and we have various other projects on top of that. Okay, so how fast can you actually process the PDB interactively? And this was actually run on CyVerse on a virtual machine with 12 cores. So the green line here, that represents just reading 150,000 PDB structures, and using 12 cores, we can easily go down to about 1 minute. We can read the whole protein data bank in 1 minute. And then, for example, to find all zinc interactions, I talked before, the example, maybe takes about 2 to 3 minutes. So basically means we can do large-scale data mining of the protein data bank, in an almost interactive session here. So far we talked about 3D structures, but we really need to see 3D structures in a biological context. So we need to integrate with sequence information, with protein function information, with ligands and drug molecules, maybe even with literature. So we need to find a way to integrate with diverse data resources. So I'll talk a little bit about that. What we do, we take data from various resources and standardize their data in a format that's suitable for parallel processing, and that's called data frames. And I will explain a little bit more about what those are. Once we have those in place, then we can run, for example, 3D structural analysis. We can integrate the data with other biological data. We can create data sets for machine learning. Many people now start using machine learning and structural biology and so forth. So in a traditional way, many data resources consist about many small files, and that's obviously a problem. And those are usually custom file formats. So you need a custom file reader, you need custom data structure, and you need custom applications. So this approach, doesn't really scale well. So to work with Spark to process things in parallel, we need to have more structured data formats. So for example, we could have data in a comma-separated value file or in XML or JSON. But to really have a high performance, the interactive performance that we are looking for, we really need high performance file formats, and we chose Parquet and org that gives the highest performance is Parquet and org. So Parquet and org are so-called Columnar storage formats. And basically the idea is on the left-hand side here, there's a table of data. And usually this is represented as records in a database or records in a file. But what Parquet and org does, they represent the data as columns in a columnar fashion, and that provides some space efficiency as well as increased query performance. So if you have a database that contains data about animals, and let's say you only need the cat data, you can pull out a column of cats, for example, very easily. So using those storage formats, we can do something called predicate pushdown. That means we only need to read the data that are really required for an analysis, and that speeds up things dramatically. So to give you an example, one important data integration point for us is to integrate UniProt protein sequence data with the 3D structural data. And that mapping is maintained by the Protein Data Bank in Europe as part of their SIFTS project. And those data live in about, when I did the benchmark, in 140,000 gzip XML files. And to download them, it took me 27 hours to just download the files that contain 105 million records. Now, this is definitely not scalable, and you can't run this interactively. So the original data files together were about 5 gigabytes of data. Then we extracted a subset that we need into a comma-separated value file, but it was still about 500 megabytes. But then if we use a Parquet or org file format with some compression techniques, we can get this down to about 50 megabytes. Now that's one thing, that we reduce the size about a factor of 100. But more importantly, now we can do actually operations on those data sets. So this is 105 million records, and we can do calculations on the fly. And this benchmark was actually run on my laptop here. So for example, you can do operations on this. You can just count how many records there are in 3 or 4 seconds. Here's an example running a query that ran in about 12 seconds, or we joined some data sets. So basically you can do operations like the data set of the 100 million record size on a laptop even, interactively. So this is, an important step for us to make this, fast enough. So the way we then analyze data is we take the original data sources, we convert them, we clean them up, and so we end up with very few data files, and they are stored in those data frame file formats like Parquet or org. And then we can just use standard libraries. We don't need to write any software, and then do some data analytics on those data. So with a few lines of code, we can then analyze those data. Okay, so now we're talking a little bit about CyVerse. So how does CyVerse get into this picture? So CyVerse is an NSF-funded infrastructure for data-driven discovery. So this is a very nice fit with our project. And it's a national infrastructure for the life sciences, both for research and for training. And this webinar is one example of their training efforts. So recently, they added a component to run interactive analysis, and this is called VICE, Visual Interactive Computing Environment. And to define your software stack, you use a thing called Docker. Don't worry about it if you're not familiar with that. The people at the CyVerse team can help you set up basically a reproducible environment for your software. So when a new user comes in, they don't need to worry about software installation. It's all ready to go. And then you can expose those calculations through a web browser to the end user. So this Vice environment addresses many problems. There's no installation required. Even non-technical users can use it. There's a button style, and I'll show you that basically means with one click you can rerun an analysis. They host the data for you. They run on enterprise hardware. It's secure, and more importantly, you now can share your analysis with other researchers or the public, and they host everything for you. Okay, so back to my overview slide here. So I told you a little bit how we run a scalable analysis using effective data formats like mmtf and Parquet and org, and parallelizing them using the Spark environment. And this said, we have now set up a prototype that runs on CyVerse, and we're going to do a demo soon. So one example that we have worked on is to map data, either proteomics data such as post-translational modifications, or genomic data, for example, mutations or insertions or deletions, and map them onto a 3D structures to see where they are. And the reason why we want to do that, if you look at a protein or genome sequence, the mutations may be far apart, but if you look in 3D space, those mutations may actually cluster in a particular area and have some functional impact. So in particular, the analysis I'm showing you today, we look at mapping mutations to 3D structures and then see, do those mutations occur at protein-protein interfaces, at protein nucleic acid interfaces, or at interface between proteins and ligand or drug molecules. Okay, so as I mentioned, we have been working on mapping post-translational modifications to 3D structures. So this is one effort, but today I'm focusing on mapping mutations to structures. If you map a mutation from the gene to the protein and finally to the 3D structure, you can get some information. This is, for example, a zinc finger, and there's a mutation right at the zinc binding going on here. So this probably has, some effect on protein function. Here's another mutation. This is a protein trimer. And if you make this modification, this trimer will, for example, not form. So looking at 3D structures, we can get some additional information that you cannot get from a genome or protein sequence. Okay, so let's start with a demo here. This is our mmtf-genomics project, and this is hosted on GitHub. So let's quickly go to GitHub and let me show you what we have here. So all the source code, all the Jupyter Notebooks are hosted on GitHub. And the nice thing is you can run those yourself in your web browser using two services. One is called Binder. This is a service provided by the Jupyter team. This basically runs on the Google Cloud, but it's very limited. It only gives you like 2 gigabytes of memory, and there's really no way to store your data and so forth. Now, fortunately, we learned about Vice, and Vice solves all those problems for you because users can have accounts, they can keep up to 100 gigabytes of data, and they can share their analysis. So you can run an analysis in Vice by just clicking on this launch button. There are a few more steps, so they are described in here. You need to create an account and need to log in and so forth, but it's really very easy to rerun analysis that somebody else shared for you. So in our mmtf-genomics project, we did a number of things. We mapped mutations, for example, from the dbSNP database to 3D structures, and here you can do some filtering. For example, find all mutations that are clinically significant based on ClinVar annotation and map them onto 3D structures. Here's another example where we did the opposite. We're looking at mutations with high allele frequencies, which are most likely benign, and see where do they map onto protein structures. So today I want to focus on a pipeline where you basically have a set of mutations, the genomic location, maybe as a list of, for example, the chromosome location of your mutations here. And we can map those onto 3D structures. Let's see to get all of this. All right. So rather than launching it now, I already launched the Jupyter Notebook in Vice. So what you see here, this is what's called Jupyter Lab. So in Jupyter Lab, you have a Jupyter Notebook, and as you see, there's text in here, then there's code in here, and tabular information, as you will see as I go through the demo. You can also have 3D structural information in here. On the left-hand side, you see your notebooks are listed here. In this case, it's a 5-step process, so you see there are 5 notebooks here. You can also have data files here. So for example, in this case, we have a simple input file that just contains 3 mutations here, where we now want to know, where do they occur within a protein structure. So let's go to our second step. We now map those mutations onto a protein structure, and that is shown in here. So here's this specific mutation on chromosome 6, and if you map that to a 3D structure, this amino acid in red, that's the one that's being mutated, and we show the surrounding amino acids within 8 angstroms in this visualization. So this is an example of a notebook where we combine, text, code, and 3D visualization, and this is interactive, so you can play around with that. You see the slider up here, so you can, this data set has like 235 entries, so you can easily use those sliders to go in here. So it looks a little bit like a web page, but this thing is much more flexible. For example, if you really do programming, you can expand here. You can actually see the code here. If you want to change the way things are colored in this image, you can, just go in here, change it, and rerun this notebook again. So this is the first step. We map mutations onto 3D structures. Next one is we want to know, do they map onto protein-protein interactions? And that is shown in here. So here we have a mutation in red again, and this is at the interface between 2 different protein chains, and the interacting residues like the histidine and valine, leucine, they're highlighted in orange here. So if you make a mutation here, most likely, it will have an effect on binding. So then the next step is we want to look at, do those mutations affect ligand binding sites? And this is done in this notebook here. So in green is a small molecule that binds into a binding pocket, and right next to it in red, you see there's a mutation here. So again, we can use a slider to slide through here. We have a few other options. For example, you can also show a surface. So this is very customizable. It's not like a web page. You can, if you know how to program, you can change things around. So here you see the binding site with the ligand bound and the mutation here, right here in the binding site. So, and then people who are interested in drug development, they may want to know how does it affect drug binding, and that's done in this notebook here, where we map, again, mutations to proteins that are close to bound drugs. So in this particular example, we have a few kinase inhibitors. So those are FDA-approved drugs that I'm showing here. And we show some mutations that most likely affect drug binding and may cause drug resistance. And again, using the slider here, you can slide through the various examples here. Okay. Yeah, and important thing about this is that you can change the code, and it's completely reusable. Let's say you have your own data sets of mutations. You could go in here and basically read your table in. So what we're doing here, we're basically reading a file, in this case a tab-separated value file in, but if you have a file of mutations, you just need to change this here and read it in, and then you can go through this process yourself. So this is the idea of reusable code, and again, you can change things around too. Now those notebooks run really quickly because they use this mmtf file format, and it's parallelized to Spark. And finally, we represent big data through effective file forms such as org and Parquet. All right. So that's kind of my demo. Let's see, how do we look in time? So we're just about at the end here. So let's go back to my slides here. Okay, so if you want to get involved with that, what can you do? If you're if you have your own Jupyter Notebooks applications you want to run in BIOS, there's some very nice documentation available here at CyVerse.org. And again, the CyVerse team is extremely helpful. So they helped me a lot to get set up here. So I highly recommend working with the CyVerse team. If you want to know more about our project, we have an entire 3D workshop, which is online. This is the workshop we had last year here. So there are many materials, about 100 Jupyter notebooks doing all kinds of different type of analysis. If you want to know more about it, you may want to work through this notebook. This is not available on CyVerse yet, but hopefully next week we will deploy this also on CyVerse. So you can just run this entire workshop in CyVerse. If you want to know more about our own project, there are various links in here. If you want to run mmtf-genomics, the link is here. I didn't talk about our proteomics project. Again, if it's available, you can run this as well here. And what we're really looking for is for you to get involved. So first of all, if you have any help, feel free to contact me. If you have any ideas, kind of analysis you want to do, I'm really looking for what kind of new features should we add next, or if you're interested in some kind of collaboration, I would be very interested in that as well. Finally, I want to acknowledge a number of people. First, I want to acknowledge the 3 PDB teams here at the RCSB PDB, the Protein Data Bank Europe and Protein Data Bank Japan. Actually, you didn't realize it, but as I did my analysis, we are using data from actually all 3 resources in this notebook. I want to thank Mars Huang, who actually wrote most of the Python code here. And I want to thank Lorne Fan from the NCBI, who helped us with the dbSNP data integration. I want to thank JJ Gao and Shujing Wang, who helped with the genome to structure mapping. And then, of course, want to thank the CyVerse team, especially Nirav, especially Upendra, who really helped me get started, and many of the other team members here. This was funded by an NIH NCI award. And finally, CyVerse is funded by the National Science Foundation. Okay, that's my presentation. Any questions?
Yes, Peter, there are some questions in the chat window, so I'll read those. Can the Docker images be run on other non-CyVerse servers without additional changes?
No, so those are specific. But what we have, if you go to our GitHub repositories, we have a complete setup within 2 or 3 steps. What we do on other environments, if you want to install this software on another server or on your laptop, what we provide is basically a Conda environment. You install a Conda environment. It's also it's not a Docker image, but it's a reproducible environment that you can easily install with about 5 commands. So it's very easy. We don't have a generic Docker image yet, but that may be something we consider down the line.
Okay. That's the only question I see. Are there any other questions for Peter, since we have quite a bit of time left if we need? Okay. Is there an RNA tool?
What is that, an RNA tool? I'm not sure what that is.
Yeah. Oh, I'm sorry. It came to me privately, so let's see. Is there an RNA tool, I guess, in terms of what you've provided here?
Yeah, I'm not sure what an RNA tool is supposed to be.
So maybe the person who typed in that question can unmute themselves and ask more specifically what you're interested in. I think you can unmute yourself. Unmute. Meg? Anyway, okay, well that's not. Peter, let's see. Okay, there's another question. Have you tried using MATLAB scripts within the CyVerse environment?
No, I have not, but I think there was some discussions. There's this Octavia tool which basically allows you to run MATLAB scripts, in an environment. And I'm not sure if the Vice team would need to address that question, but I know that, for example, Binder supports to some extent running those type of scripts as well.
Okay.
The problem with MATLAB is that a license is required. Therefore, you cannot just publicly provide that kind of service. So then open source alternatives like Octavi could be used for that.
Okay. There's a little bit more information now about the question about the RNA tool. The person doesn't have a mic, so it says you model RNA enzymes.
Oh, I see. Yeah, right. Although it's called the protein data bank, it also contains all the nucleic acids, including RNA and DNA. So that same analysis can obviously also done on RNA and DNA as well.
Okay, I think you've answered the question then. Are there any other questions for Peter? Are there any user security issues using Jupyter Notebook and covers?
Yeah, I'm not really an expert on this. I mean, the CyVerse environment, the way you launch Jupyter Notebook is through HTTPS. So this is, encrypted. So, it's safe, but, I'm not really involved with proprietary data or, sensitive data. So I can't really answer that question.
Nirav is on, and I'm not sure, Nirav, if you want to take a stab at the security issue, but or concern.
So I'm not sure what specific insecurity they're looking to, but in terms of your notebook, it's private. Nobody else can get to it. You have to go through authentication. So it's not an open notebook, but you are allowed to share it, so you can invite somebody else to join you in the analysis if you may. But if there are specific security questions, I'm happy to answer them if you can go deeper into it.
Okay.
Maybe if there are no more questions, one thing I could try here to actually show you how you run this live with a one-click launch. It's always so dangerous to do a live demo, but if there's interest, I could quickly try this.
Sure. Clearly we told people it's about a half an hour, so anyone is welcome to stay as you demonstrate further. Otherwise, some people may leave, but go ahead, Peter.
Yeah, okay. So on this mmtf-genomics page, you see this link, Launch By. So if you click on it, you will get into the Vice environment. And basically you get to this page, and to actually launch the analysis, you need to click this button, Launch Analysis. So it's really almost a one-step, to get to a running notebook and to run any of the analysis that I showed you. It says up here, it successfully launched it, so we need to wait a minute until it basically sets up a virtual machine. It takes that Docker image and actually creates a Docker container. It sets up the entire environment on this virtual machine, and then when it's ready, we can try to access it. So I click on here, and then you see the Jupyter Lab launchers, and then what I showed you is in this folder, mmtf-genomics pipeline 1, you see here my Jupyter Notebook, so the first one. I open it up, and then I go to run all cells. So this basically, now it's actually running as we speak. It just ran the first step, and then I'm going to the second step here, and now map those mutations onto 3D structures. Again, you could provide your own mutations as well, and rerun this notebook. And again, let's just run this step just to give you an idea how fast this is. I mean, this is just a few mutations, but just to give you an idea, So this is basically within 2 clicks and a login, you're basically right there in a living notebook. And again, if you're a programmer, if you know a little bit about Python, you can go in here and actually change things around. As you see, it's still running right now. So in a few seconds, you will see the visualization on the bottom. So actually, this is interesting. This particular step, which is running right now, that is actually downloading those about 100 million records of PDB uniprot mapping. So that just happened right here, and it's still running. So this will finish. in a few seconds or so. So here now we see how the mutations are mapped onto 3D structure. So this is how easy it is to, from a one button click to get to this place that I showed in my demo here. All right, so that's all I have to say. So again, we're looking for collaborators. My email is in this presentation, so feel free to contact me. I'm happy to work with you through some use cases.
Okay, if there's no further questions for Peter, we'll call it the end of the webinar. Thank you so much, Peter. That was really helpful, and I think you've given our community a great, another resource to use on their science. And thank you all for attending today's webinar. Please be sure to join us in 2 weeks for the next Focus Forum presented by Upendra on doing metagenomics with QIIME 2 using Jupyter Notebooks and Vice. And have a great weekend.